Thank you, George. I appreciate being here. I appreciate the invitation. And I was asked to talk about, in general, about designing an RSV vaccine. So these show my disclosures. As George mentioned, we have a CRADA between my lab and MedImmune to develop live attenuated RSV strains. And our lab is also developing RSV vaccines based on parainfluenza virus vectors. So you've heard about RSV already today. It's the most important viral agent of pediatric respiratory tract disease worldwide. It's a paramyxovirus. It's an envelope virus with a negative sense genome that we sequenced back in the 1980s. There's a single serotype, and there are two neutralization antigens, which are the major protective antigens. These are the F and G glycoproteins, and F is more potent and more highly conserved. So RSV is best known as a pediatric pathogen. It infects efficiently very early in life, so that by the end of the second year of life, essentially everyone worldwide has been infected. And it has a substantial impact. In children under 5 years of age in the United States each year, 1 in 10 sees the doctor for RSV disease. That's each year. And there's substantial hospitalizations, but with supportive care, the fatality rate is relatively low. But worldwide, there's substantial morbidity and mortality due to less supportive care. There are estimates up to 1 million deaths per year for RSV. In addition to the acute infection, severe RSV disease early in infancy can be followed by pulmonary function abnormalities that last into adolescence, and RSV has also been controversially linked to the initiation of asthma. In addition, RSV has substantial impact in the adult population, in severely immunosuppressed and elderly populations in particular, and its fatality rate and impact in those populations approaches that of influenza. And RSV remains largely uncontrolled. There's this passive immunoprophylaxis with the palivizumab antibody, which gets substantial use and reduces hospitalization substantially. But it's expensive and it's not available for the general population. We lack an effective antiviral against RSV, and we lack an effective vaccine. And we generally think about two types of vaccines. One would be a pediatric vaccine for RSV-naive infants given early in life, and a second would be an adult vaccine that would be given to individuals that by definition are adults who have already been infected with RSV, so it would be a booster vaccine. And that would have several uses. One, for example, would be maternal immunization, which is a strategy to boost the level of antibodies in expectant mothers so that the newborns will have higher levels of RSV antibodies. And this has been pioneered with influenza, tetanus, and pertussis. The elderly would be another population for vaccine use, and also individuals with underlying disease. So this is a vaccine snapshot that I borrowed from the PATH website and modified slightly. And it shows current activity for RSV vaccine development. So this is a time when there's a considerable new interest in RSV vaccine development. And this shows vaccines that are in preclinical phase 1 and phase 2 studies. And there are three groups of vaccines. The first on the upper left is live attenuated RSV strains. The second group are vectors for RSV vaccines. And these include parainfluenza virus vectors, other viral vectors such as alphavirus, adenovirus, and MVA, vaccinia, and also nucleic acid virus vectors. And then the third group at the bottom are protein vaccines, and these include inactivated whole virus, particle vaccines, and also subunit vaccines. And you can see most of the activity is still in preclinical. These boxes are too small to see, but each one's a different vaccine initiative. But you can see some that have advanced to clinical studies. In the upper right, the live attenuated strains, there have been at least 6 strains evaluated clinically over the past 15 years. Moving down, there's been a parainfluenza virus vectored vaccine that's been evaluated. And further down is a Novavax particle vaccine made of F protein that Greg Glenn will talk about in the next talk. So RSV vaccine development has been underway for almost 50 years and has been a tough goal to meet. And these are some of the obstacles. So immunization should begin early in childhood, early in infancy, in fact, the first few months of life. And safety in this population requires stepwise testing going from adults down to younger infants. Immune responses in infancy are reduced due to immunologic immaturity and immunosuppression from maternal antibodies. In addition, vaccination in this time would overlap with some illnesses of infancy, such as apnea and SIDS and so forth, and there could be a perception of a link to that to RSV vaccines. The protection that's conferred by natural infection is incomplete, and so we would expect that might be the same for RSV vaccines. RSV grows inefficiently in vitro, and it's difficult to handle. That's another obstacle. And finally, there's a lack of convenient experimental animals. So we can use cotton rats and mice and African green monkeys and so forth, but they're all semi-permissive. And chimps are no longer available. And this is perhaps the biggest obstacle, which is the disease enhancement that's associated with certain RSV vaccines. This was first observed in this well-known episode with a formalin inactivated vaccine that was evaluated in 1966 in a number of field trials. And this was done in infants and children. And it turned out to be poorly protective. And in addition, when those individuals were infected, they suffered enhanced disease, so that in the youngest age group, 80% of the vaccinees were hospitalized, and 1 in 10 died of RSV disease. This phenomena has been reproduced to some extent in experimental animals, but it's not clear how reliable those models are. And a number of factors appear to be involved. So poor antibody responses, poorly functional antibodies, poor TLR activation, poor CTL responses, complement activation, tissue damage, and a Th2 biased response. So it's a complex phenomena, and it basically is thought to involve antigen presentation issues. So who's at risk? This is specific to RSV naive individuals. So it's associated with the first exposure to RSV antigen in life. So it's an issue for pediatric vaccines. It's not observed in individuals who have been previously infected with RSV, so it's not an issue for an adult vaccine. And which compositions? Well, it's observed with formalin inactivated RSV, as I've mentioned, also with subunit vaccines of F or F and G. So it seems to be an issue for protein vaccines in general for RSV. But it's not observed with live attenuated RSV or wild-type RSV in nature, or with vectored RSV vaccines. And so this has been shown both in experimental animals, and it's also been confirmed clinically in clinical studies post-vaccination surveillance, both in studies of live RSV vaccines and in studies of vectored vaccines. So those are the vaccines, the only vaccines for RSV that have been shown to be safe in young infants. So this now is the vaccine, returning to the vaccine snapshot, and I'll briefly summarize a few different vaccines. So this is first looking at live attenuated RSV strains. So these are produced by introducing attenuating mutations into live RSV. This is done using reverse genetics, so it's done making virus from cDNA. This provides virus which is clean, has a short passage history, has well-defined mutations. It's intended to be given during the first months of life by the intranasal route. So the advantage of immunizing intranasally with live virus is one gets broad stimulation of innate and cellular and humoral immunity. One, of course, also gets mucosal immunity in the respiratory tract. All the antigens are displayed to the immune system. A single dose is substantially immunogenic. Studies with influenza virus vaccine have shown that the live vaccines are more immunogenic than subunits in RSV naive individuals. And as I've said, this is free of disease enhancement. But there are drawbacks. It's an infectious agent, so one must attenuate, and that's been the biggest stumbling block. It's a lengthy process. There's also the fear that in some individuals that the live intranasal vaccines may have some residual reactogenicity in a small fraction of the population. And also there's a difficulty of working with the virus because of its poor growth and instability. A number of years ago, we reported on a promising live attenuated strain. It has a complicated name up there, but we also call it now MIDI 559, and has a series of mutations that I can't point to, but they include a number of point mutations collected from 4 different viruses, also deletion of this small gene called SH. And this produces a highly temperature-sensitive virus, and so it's a safety feature because it's restricted in the warmer, lower respiratory tract. So this was tested in a phase 1 study and all the way down to RSV naive 1 to 2 month old infants, and it was well tolerated. It was moderately immunogenic. It gave substantial restriction of a second vaccine dose, indicating there was protective immunity in most individuals. There was some genetic instability, so we observed a shift in some virus isolates, a temperature shift, and we identified reversion at one of two sites. These partial revertant viruses remained highly attenuated, and there was no increase in illness. And so clinical evaluation of this vaccine is continuing, but ideally we should have increased stability. So we worked on this virus to stabilize those 2 point mutations, which are boxed there, and we've been able to increase their stability, and we have this new virus called CPS2 that we're going to be analyzing this spring. We also have other candidates that are moving into the clinic this year. So this virus involves deletion of that open reading frame over there on the top in the middle called M22, which encodes an RNA regulatory protein. So when we delete this, we get increased transcription and increased antigen synthesis, and that could be seen in the box to the right, which is a Western blot, comparing synthesis of F protein by the wild-type virus on the top versus the mutant on the bottom, showing increased synthesis of F protein. So this may give increased immunogenicity. We'll have to see. This virus also should be genetically stable because it involves deletion of a whole ORF. And this is presently in phase 1 studies, and those should be finished in a few months. This is in seronegative infants. We have a second virus, which is moving into the clinic this year, which involves deletion of a codon in the L polymerase gene, which is over on the right in red, and also deletion of an interferon antagonist gene, which is on the left. This should also give increased immunogenicity because of the adjuvant effects of interferon. It should be stable because of the deletions, and we're beginning evaluation this year. And so from these new candidates, we're using our increasing knowledge of RSV biology to design what we think would be improved viruses with improved features of immunogenicity, genetic stability, and so forth. Okay, this is returning to the vaccine snapshot and now looking at vectored vaccines. So the Paraflu, which is the most advanced, the other viruses I've mentioned, and nucleic acids, although I won't talk about that today. And these vectors can be both replication competent or they can be replication defective, depending on the vector. So these also have the advantage of being free of disease enhancement. They avoid the difficulties of working with RSV, which is a big advantage, particularly moving into resource-challenged settings. They have a disadvantage, though, that they express only a single or at most 2 RSV antigens, so you don't get the full repertoire of immunity against RSV. So the Paraflu vectors are the most advanced. Both our laboratory and MedImmune are working with the Paraflu serotypes 1, 2, and 3, and St. Jude's is working with Sendai virus. And so these are complete replication-competent viruses, and they have the F gene added as an extra gene, and it can be placed in several places depending on level of expression that's desired. And so one advantage in particular for the PIVs, or human PIVs, are these are very common ubiquitous pathogens of infancy, and so these are not something, we're not exposing the infant to anything new. And in fact, we're providing them with a bivalent vaccine against the PIV and against RSV. And all these PIVs are viruses for which vaccines are needed, particularly PIV3. This strategy is similar to the live attenuated vaccine strategy in terms of giving a broad stimulation of immunity, including mucosal immunity. But on the other hand, they're infectious agents, they need to be attenuated. But we do have attenuated versions of these viruses in clinical studies. And in initial studies, preclinically, these vectors look very good in rodents and non-human primates. So one of these has advanced into the clinic. This was done by MedImmune. And this is a vector that's a chimeric vector for PIV3. So the internal genes are from bovine PIV3, the ones that are in brown, and those confer host range attenuation in primates. The genes in blue are the major neutralization antigens. Those are from human PIV3. And then the F gene is added between the N and the P genes as an added gene. So this has been evaluated and reported in infants and children 6 months and older by MedImmune, and they have a further study that they haven't released yet. So in the published study, this vaccine was well tolerated. It was immunogenic against PIV3, somewhat less immunogenic against RSV. There is some evidence of instability in the F insert, so that would be something that needs to be monitored. And we're working now to see if we can increase the level of F protein immunity that's induced by this vaccine. This is an example of another type of vaccine for RSV vaccines, another type of vector, adenovirus. And we actually worked with Wyeth in the 1980s with replication-competent adenovirus with a number of serotypes. And we evaluated them in cotton rats and dogs, and they looked very good. They're very immunogenic and highly protective. But in chimpanzees, they were poorly immunogenic. So we discontinued using these. In the meantime, there's been a tremendous amount of activity developing adenovirus vectors for gene therapy applications. And so now there are new generations of vectors available for RSV vaccine work. And these involve deletions in various genes. And they also involve new types of adenoviruses, such as gorilla and chimpanzee adenoviruses that are antigenically unrelated to human adenoviruses, and therefore they're not going to be inhibited by a pre-existing antibody in humans. And so those need to move into the clinic. The last part of the vaccine snapshot are protein vaccines, and those are inactivated whole virus, different particle vaccines, and then subunit vaccines. And these have a tremendous advantage of being non-infectious. That's a tremendous advantage for manufacture, for storage, for use, for safety. On the other hand, it's common for protein vaccines compared to live vaccines to have reduced repertoire of immune responses, in particular reduced innate and CTL responses. And there's also the history of disease enhancement that I've mentioned for pediatric use. So work with these protein vaccines began in the 1980s and 1990s with purified F protein from RSV-infected cells, and a variety of clinical trials, including maternal immunization. The immunogenicity was disappointing. There was then this subsequent vaccine of F, G, and M that Sanofi made, and the immunogenicity for that seemed to be somewhat better, but this has been discontinued. Now there's a new vaccines coming available. There's the particle vaccine by Novavax, which Greg Glenn will talk about. There's also other particle vaccines, VLPs and virusomes. There's also new forms of engineered F which are being developed. And there's a cartoon over on the right that shows the pre and the post-fusion forms of F. The pre-fusion form of F is the one that's found in the virus particle. When that contacts the host cell membrane, it gets triggered so that heptad repeats, the green things that say HR, those swing down to contact the blue HR domains, and that brings the membranes together for fusion, and it gives the post-fusion form. So a number of laboratories are working on stabilizing both the pre-fusion and the post-fusion form as new improved antigens. And so 2 years ago, 2 laboratories reported on a stabilized post-fusion form, and those are presumably now moving into the clinic. And laboratories are also working on the pre-fusion form. And the pre-fusion form is particularly interesting because it appears to have a number of neutralization antigen epitopes that are unique, that are major epitopes in for antibodies in post-infection serum. So that's a very interesting, very exciting line of experimentation. So to summarize, I've mentioned that we need to have 2 vaccines. One's a pediatric vaccine and one's an adult vaccine. So for a pediatric vaccine, with the live attenuated strains, we put tremendous work into that. We have a number of strains that seem to be suitably attenuated. We've improved their design to make them more immunogenic, we think, and more stable. We now have to see if they're protective, and so we need to do efficacy studies with a lead candidate. We also have the PIV vectored vaccines, which have been in the clinic, but which we think we need to improve the RSV immune response. And then there's the questions of the other vectors, the adeno and the alpha virus vectors, how will they perform in humans? That needs to be determined. But all of these have the advantage of being free of disease enhancement. In contrast with protein-based vaccines, it's not clear if these will be safe in RSV-naive recipients. It's not clear what kind of preclinical data would be convincing that they're safe. But one possibility is that we could use them in a prime boost, where we prime with a live attenuated vaccine and then boost with a protein vaccine, and they should be fine in that application. Now, the adult vaccine, we know from our own studies that the live attenuated strains are too restricted and too attenuated to be suitable, so we can't use those. The same is going to be true of vectored vaccines that are based on human pathogens. Pre-existing immunity in humans is going to make them too restricted. Vectors like the non-human adenoviruses, those could be used, and we need to see how they perform. And of course, protein vaccines would offer tremendous advantages for use in RSV-experienced individuals They'll be less sensitive to pre-existing antibodies. They could be given in multiple doses. And we're hoping that new developments in antigen preparation will yield improved vaccines. So finally, the pediatric vaccine, we have the live strains, we have the PIV vectored strains, we have vectors such as adeno, which remain to be tested clinically. We have the continuing question about protein vaccines. Can they be made suitable for infants? We have the adult vaccine, which has potential for vectors such as these non-human adenoviruses or alphaviruses. And protein vaccines have tremendous potential in this population. And so in conclusion, I'd like to thank members of my own group at NIAID, also our clinical study site, Johns Hopkins, Wyeth, our previous collaborators at Wyeth, which is now Pfizer, and also our present collaborators at MedImmune Vaccines. Thank you.